Hello. In today's class, we're going to be talking about an overview and introduction of Nextflow and containers. We have three goals in today's class. First is just to introduce Nextflow and talk about the advantages of using Nextflow or a similar workflow manager over just a series of custom bash scripts. Then we're going to look at some Nextflow code just to get an idea of what the syntax of Nextflow looks like. Although we're not going to be programming any Nextflow in this course, it's good to get an idea of how to read Nextflow code. And finally, we'll talk about containers and how they pair with Nextflow to create a fully reproducible workflow. And we'll talk about the differences between Singularity and Docker, which are two main container engines. So first, let's start with why we might want to do reproducible research and put a lot of effort into making our computational analyses reproducible. And the first is to show the correctness of your results. Generally scientists are skeptical people, and there are those in your field that may want to follow up on your results or build on your results, and being able to reproduce them is a really good first step in getting your results out there and letting other people verify them. Without having a working piece of code, people may struggle to understand what exactly it is you've done. It's hard to write a method section that is detailed enough to actually implement the exact same thing in code. And so really the code is, in a way, the methods section. And you want to avoid this picture on the right here where two scientists are talking and the main explanatory step is then a miracle occurs here. So basically that's what we're trying to avoid. And one of the principles driving reproducible research is this idea that really the article itself is the tip of the iceberg, and that's advertising, not scholarship. The actual scholarship is the full software environment, code and data that produces the result. So really, we want to be delivering the full package, the full iceberg, when we publish. And you'll notice here, it doesn't just say code, it also says the full software environment. And why might that be the case? Like, why is code not enough? And the answer is because often the software packages that we're using in computational biology are quite complex. So this is a dependency graph of a piece of software that's not used in computational biology, but it is common in its complexity for a lot of the softwares that we use. So this is actually for a web services framework. But the principle is there that often, since these softwares are so complex, you really need to have the exact version running on the same operating system in order to get the same result. And just to continue on this point of why code is not enough, there is a really great publication from 2013 talking about how long it takes to reproduce a computational biology paper. So in this case, they focused on one paper, and this was a paper that provided all code and all the supporting data that was required to run the computations was publicly available. And it still took experienced computational biologists on the order of 280 hours to reproduce the results of this paper from end to end. So that's 7 weeks of 40-hour work weeks, which is a huge undertaking. And really, we can do better than that with modern technologies like Nextflow and containers. And the goal really is to be able to reproduce a workflow using one command that even a novice can run, and then it will reproduce all of your results starting from the raw data all the way to the final figures. So really, there is, of course, a spectrum of reproducibility, whereas on the left here, if you just have a scientific article, the publication only with no code, that's obviously not reproducible. But having just the scripts available, it's really not that much better. And for full replication, for the gold standard, you want to have the linked executables, all the software that you used, and all the code and all the data available. So that's really what we're aiming at. I want to show you now an example of a really good gold standard reproducible piece of computational research. So this is from Casey Green's lab at UPenn. This paper is really one of the best examples I've found that has all of the hallmarks of reproducibility. So just to go through what those are, it uses code instead of pointing and clicking. That means that there are no gaps or steps where maybe code is used for the first part, and then there's a point and click step, and then code is used for the next part. The whole thing, from end to end, from the raw data to the final figures, it uses code. And secondly, it's open source. So you can see on that previous slide, I showed you the GitHub, and code transparency and having all of the code that you wrote open source is key to reproducibility. It's also got version tracking, and in particular, the versions of software that are used are noted, and the analyses themselves are documented in Jupyter Notebooks or R Markdown. And we'll talk more later in the course about what R Markdown is and why that's valuable to use. The data is archived and available, and the provenance of that data is well documented. And if there is any stochasticity in the data, pseudo-random variables have a seed set so that you achieve the same result each time. And the software environment is also replicated using a container system like Singularity or Docker. And finally, it's best if the workflow is automated so that someone who would like to reproduce this work can run it with a really simple command. And in this case, in order to do that, we can use tools like SnakeMake, Common Workflow Language, and Nextflow. So the really cool thing about this paper called BioBombi from the Casey Green Lab is that it does all of these things. So this is a great gold standard to look at and study if you're looking to make reproducible scholarship yourself. So let's talk about some of the challenges that arise, in particular in computational biology, in trying to develop reproducible workflows. One is that these workflows can be quite large, and they're also embarrassingly parallel, which means that maybe each FASTQ or each experiment can be run separately for most of the computations that need to be done on that data, and that there can be hundreds or sometimes hundreds of thousands of samples that you're analyzing. And there's often many different tools, languages, and scripts that come together to make a computational biology workflow, which means just one language is not flexible enough to capture all of that complexity. And finally, there are really complex dependency trees and configurations, which makes it a very fragile software stack. It could be the case that just changing one parameter, such as for Chip-seq, parameters having to do with peak calling, can change the results quite a lot. An example of how results can change and how having such a fragile software stack makes some results irreproducible comes from the Nextflow publication itself. So the publication that you cite if you're using Nextflow is that they ran the same gene annotation pipeline on a few different operating systems. So this is a pipeline with the same software, the same versions of that software, but purely just on a different operating system. And you can see on the left here that we have most of the genes in common, but really there are 85 genes that are unique that you get when you run it on a Mac OS, and 3 unique genes when running it with Amazon Linux. And that is concerning because that is really, everything else is the same, all the software versions are the same, and it's just the operating system that's different. So similarly, over here, this is a different pipeline. So this is with transcript quantification, and you can see that really on Amazon Linux, you're getting unique significant genes, and on Mac OS, you're getting unique significant genes. And really, this is what we're aiming at is full reproducibility, where it doesn't matter what computational environment you're running in, that you get the same differentially expressed genes, you get the same answer each time. And Docker, in this case, is what solves that. So this is using Nextflow plus Docker. You can see that when it's run on Amazon Linux or Mac OS, that you get the same genes differentially expressed. So really, that's what we're aiming at, and that's one of the things that Nextflow can offer us as a workflow manager. So now let's talk about why we need a workflow manager. That was sort of a tease of the motivation, but what does a workflow manager do, and why do we need it? So with a workflow manager, you specify explicitly all the data and scripts needed to run your computational analyses from end to end. And Nextflow abstracts the execution environment, which means that you can run it locally on your machine, or you can run it on a compute cluster, like we'll be running it on Fiji, and you really don't need to change much to make that happen. So what's really cool is that all of the business logic, as it were, all of the logic that says which steps will be taken to analyze the data is totally abstracted from the parameters and settings to tell it what environment it will be running on. And finally, Nextflow is really fast to develop because of the way that they have engineered the language. Another really nice feature of Nextflow is that there is caching at every step of the pipeline, which means that if you need to rerun the analysis for some reason with new data, you don't have to start all over again, and it will basically check to see which files need to be recomputed and which will be changed, and which you can just use the cached version, which really speeds things up when some of these workflows take multiple days or weeks to run. So Nextflow is a reactive workflow framework and programming domain-specific language that eases the writing of data-intensive computational pipelines. And it's designed around Linux and the idea that really Linux and Bash is like the common language of data science. And therefore, since we've been learning about Bash, it'll be easy for us to understand what Nextflow is doing. So there are a few principles behind Nextflow that I want to highlight. One of them is that it enables fast prototyping with a domain-specific language, but it also is built on Groovy and Java, which means that if there are more complicated features that you want to build, you're able to do so. And it also has this idea of implicit parallelization, where things are parallelized when it makes sense to parallelize them. We'll talk a bit more about that later on. And finally, it uses this component level approach where each task execution is idempotent. Idempotence basically means that if you run the same operation more than once, you get the same result each time. And finally, it has this concept of portable deployments. And this is what I was talking about when I was talking about that abstracted execution layer and the fact that the configuration for what system you're running it on, whether it's locally or in the cloud or on a compute cluster, is separate from the logic of the analysis workflow. So really, to summarize the concept of what Nextflow is, it is a workflow manager that allows you to orchestrate complex jobs and parallelize them. And it does so in a way that is scalable and portable. You can run it on AWS or Google Cloud or Slurm, like we will be running it on with Slurm on Fiji. And then it also plays really nicely with containers, which allows you to have it be reproducible, and also plays nicely with GitHub and Git and all of these other version control systems that also allow you to do versioning and have that be reproducible as well. So really, I have this here just for reference. It's good to know that Nextflow is not the only workflow management software. Galaxy, Toil, SnakeMake, and BPipe are other popular ones. And this is a comparison of the features of each of those. But really, in this course, we'll primarily be focused on Nextflow and not really talk much more about any of these other pipelining tools. So that's, in summary, a brief introduction to Nextflow. Now let's take a look at some Nextflow code. Okay, so Nextflow's basic units are processes. And this is an example of a process which will take FastQ files and run FastQC. And FastQC basically just gives us some QC plots and information on the reads in the FastQ file. So breaking this down piece by piece, first we'll take a look at really the meat of the process, the action that happens, which is down here in the script area of this process. And anything enclosed within these three quotes is just bash script, a bash shell command that tells what this process will do. So this is the command that you would run if you were just running it on the command line outside of Nextflow. And then for each process, we also have to tell it what the input is. So the input in this case is called reads for FastQC, and that input will be a channel that contains FastQ files. And then you can see that for the output, we're taking anything that corresponds to star.zip, so anything that ends in a zip file, and we're going to make that into a new channel called FastQC. And finally, up at the top here, you can see that we have some parameters that determine how this FastQC process will be run. So in particular, we have the max forks parameter set to 1. Max forks determines how many processes of FastQC can be run simultaneously. So setting max forks to run means that this won't be parallelized. This will run one FastQC process, and after that finishes, it will submit another. And it tells it that we'd like to publish the results, so any .zip files that we find, we'll publish them in results/FastQC. And so just to show you how we create that reads for FastQC channel, this syntax at the top here is how to create a channel from some data in a directory. So the command is channel.fromPath, and then the cool thing is it allows these wildcards to specify lots of different FastQs at once, and that is also where implicit parallelism comes in. So if we have, let's say, 10 FastQ files in this data directory, then this would run 10 FastQC processes at the same time. However, that would only work if we set max forks to 10. So this is what a process looks like in Nextflow. And as I said, the processes and the logic for analysis is separate from the parameters that tell it what the execution environment is. So next we're going to take a look at how we tell Nextflow about the execution environment. In this case, we'd like to tell Nextflow about Slurm. So we write just these few lines in this file called nextflow.config, and inside this process parameter with curly braces, we tell it that the executor will be Slurm, and that we're going to submit jobs to the short queue, and each job will request 16 gigabytes of memory, and here we're changing the max forks to 10. Finally, at the end of this file, we just tell it that we're going to be using Singularity and give it a path to the Singularity container, which is a good segue into what I'm going to talk about next, which is a brief introduction to containers. Okay, so a container is a way of virtualizing an operating system to enable software to run predictably across systems. And they basically run on top of the host operating system. It shares the host kernel, and they're really much lighter than virtual machines, on the order of megabytes or a gigabyte versus 20 gigabytes for a virtual machine. And often a container instance is built to contain one application. So in our case, we'll be using containers that are built specifically for Chip-seq, and they have all of the software needed to run Chip-seq analyses and nothing extra. So this image illustrates the differences between a container and a virtual machine. But really, it also gives you an idea of what a container is doing. So a container might have, this app installed, let's say Max for peak calling. That software will interface with the container engine, in our case, Singularity, and then Singularity will interface with the operating system, which will coordinate with the hardware to execute. Whereas in a virtual machine, there's a hypervisor layer and a guest operating system that the containers lack. So that's why they're a bit lighter weight, and they've really taken over as the way to make large and complex software analyses reproducible. Okay, so there are really two popular container systems that you'll hear about. One is Docker, which is much more popular, and the other is Singularity, which is popular in academic settings. And I'll talk about why on the next slide. But one nice aspect of both of these is that there are a number of pre-built containers. As you can see, there's a Singularity Hub website and a Docker Hub website here, where you can search for containers that other people have built. And I've highlighted here the fact that NF-core has Docker containers, this one for rna-seq. Okay, so why is Singularity a bit more popular in academic settings? And what it really comes down to is security. So Singularity was designed to use in shared compute environments like the one that we have here at BioFrontiers. And it's easier to manage security of Singularity because it was designed to have untrusted users running untrusted containers. So that only the absolute minimum level of permissions are granted, and it just makes it easier to ensure that proper security is in place over Docker. So since we'll be using Singularity, I'll show you an example of what it looks like to create a container. A container is specified by just using a script which says what operating system is going to be used and what software is installed. So on the right is a Singularity recipe. You can see that what's going to run when this container is run is just echo roar, and it will also tell you that it's received some arguments, whatever arguments you put in. You can specify environment variables, and you can label it, that's very helpful, and version it. And there's also some data that you can put into a Singularity container, although it's recommended that any large data you host elsewhere. So basically, you take this text file and you build it using Singularity build, and then it will compile into a container that has .img. And you can see here that there is this sudo command, so you have to be a super user to build the Singularity container, which means that as just a user on Fiji, you can't build a Singularity container, but you can run a Singularity container. And so you do need to be a trusted user to compile a container. So one helpful task that you might want to do when encountering a Singularity container that you want to use is to know what its recipe file was. So in order to retrieve this text on the right from a compiled Singularity container. And so I've just put this here for reference as a way to do that. If the container is imagemagic 7.0.img, you can use the command Singularity shell imagemagic.img, and then you can just print out this the recipe file, which is saved at this location. Okay, so let's say you have a container that you'd like to use. It's already been compiled. What does it look like to run a container? So on Fiji, we'll have to first use module load to load Singularity, and then you can use a command Singularity exec, and then the name of the Singularity container here, and then whatever command you'd like to use. So when you run this command, you're running grep, but you're running it inside the DeepTools Singularity container. And to access the container in an interactive way, you can use the command Singularity shell, and then the name of the container, and then you're inside the container running a shell, and you can use any of the softwares that are installed in that container. Finally, I'd like to highlight the resource that we'll be using for the rest of this course, which is called NF-core. So NF-core has a large collection of community-based standardized pipelines to do a bunch of popular computational biology tasks. And we'll get quite familiar with the NF-core Chip-seq pipeline throughout the rest of this course. And that's all for today's class. Thank you. Bye.